In this video, we're going to talk about cellular communication. What are the general features of how cells communicate with one another? Now, you may be asking, well, why do I need to know communication between cells to learn physiology? Well, let's get into it. So here we are. Humans, we're trying to understand human physiology, but we really need to understand how a human body functions. So our body is, we are a multicellular organism, and our body is made up of organ systems. So as we're moving up this hierarchy, we go to simpler and simpler systems. So here's a multicellular organism, and we're made of different organ systems, and each organ system has individual organ levels, and organs are made by tissues, and a tissue is basically a group of cells. So here we are. We already discussed properties of molecules, and cells are basically made of molecules, but now we want to understand how cells communicate. Well, why is that important? Well, because if a tissue is made up of cells, and if a tissue and ultimately the organ and the organ system all need to work together to help us survive and function, then we really need to understand how cells communicate to make that happen. So we have different, as I mentioned, we have different organ systems, and our different organ systems not only communicate, the cells within each organ system not only work together as a system, but they also communicate with one another. So our nervous system is constantly talking about to the other organ systems. So our nervous system talks, the cells in our nervous system talk to cells in the muscular system, in the cardiovascular system, the endocrine system, which produces hormones that regulates our functions. They also talk to our cardiovascular system and muscular system, and our nervous system also talks to endocrine system. So all these communication happens through between cells, and really it comes down to how sense signals, how they receive them, and how they make sense of it. So there are general themes that scientists have identified with respect to how cells send signals, and how they make, when the cell receives a signal, how they make sense of it. So obviously it always starts with a signal. Now, some signals in our body are water soluble, so they just either travel within the bloodstream, if this was the bloodstream, signal that a cell somewhere else in the body, either it can travel through the blood, or it can just travel between two cells in the liquid that separates the cells. Or you could have signals that are non-soluble. So these would be hydrophobic. This would be like steroid hormones. So they usually would be bound by a particular protein carried. So this protein allows the signal to be carried in the bloodstream, because the bloodstream is water-based, and then carries the signal to the target organ, and then the signal does its thing. What happens to the signal? Well, signals that are water-soluble, they cannot pass through the plasma membrane. So there is a protein receptor sitting on the surface of the cell that identifies, let me bring my pointer. So here it is, the signal comes, and then there is a receptor here that specifically recognizes this signal. So the signal doesn't just bind and activate any cell. A cell that has the receptor for this particular signal, this is like a lock and key mechanism, they bind to each other, And then as a result of this binding, the cell is triggered to send signals to other proteins, molecules within the cell, until a response is generated in the cell. So receiving the signal by the receptor and then transmitting it, this whole term is called signal transduction. So that is generally for water soluble signals. For signaling molecules, these are all chemical molecules, whether water soluble or not. The hydrophobic chemical signals, they are hydrophobic, so they can easily pass through the plasma membrane. So very often they have a receptor inside the cell, and when that receptor receives the signal, it leads to a particular response in the cell, and very often this response tends to be activation of gene expression, but not always. So I just mentioned that cells communicate with each other through chemical signals, but there are different forms of chemical signaling in your body. The first form that we're looking at is direct. So this is direct signaling. And what happens, the communication between the two cells happens through these channels, proteins that are sitting on the membrane of both of the cells, and actually they are channels. They allow small molecules, and specifically ions, to pass through the channel, and this way the cells communicate with one another. So usually what happens is that this particular cell, which is called the signaling cell, receives a signal and generates some sort of response, and then that response travels through the rest of the cells. And this is very common in situations where cells have to work as a unison to, well, let's say muscles of your heart, where they have to, all the cells in the muscle of your heart have to beat at the same time. So this gap junction allows all the cells to coordinate their function together. So we call this the direct signaling. Then we have indirect signaling, where the signal travels from one cell to another. So the signal can travel from one cell to another through the interstitial fluid. And what is interstitial fluid? It's the fluid that is between different cells, and these are usually short distances. And this kind of signaling is called paracrine. So usually this target cell is nearby, the cell that is a target is nearby the cell that is sending the signal. So these signals diffuse, come out of one cell and diffuses to the next cell. A kind of specialized form of indirect signaling is autocrine, where the cell actually is talking to itself. So in here with paracrine, the cell is talking to its neighbors, and so is this one. But with autocrine, the cell is talking to itself. What does that mean? That means the cell, this signaling cell, is the same as the target cell. It's like the cell is regulating its own activity. Kind of like when you have a test and you're really worried, but then you talk to yourself and calm yourself down and tell yourself that things are going to be okay. So this is called autocrine. Another specialized form of indirect is when nerve cells talk to their target cells. So this is a nerve cell, and usually nerve cells have these long extensions called axons. So nerve cells receive a signal, and then they send that signal down this axon, and towards the end of the axon, which is very, very close to the target cell, the neuron releases what's called neurotransmitters. So neurotransmitters travel in a very, very, very tight space, and they just diffuse from the nerve cell to the target cell. And then we have indirect signaling, meaning that The cells aren't talking to cells that are their neighbor. You can have a cell that is sending the signal, and the signal is received by a target cell that is in a different part of your body. So the signal comes from the signaling molecule and goes into the blood, and then through the bloodstream it's delivered to the target cell. And usually when a signal enters into the bloodstream and goes to a distant location, we usually call these signaling molecules hormones. So that's why this is a hormonal signaling. Now let's get to know some major classes of receptors and how they send signals that they receive into the cell to make something happen. The first class of receptors we're going to look at are receptor ion channels, and I have a video here to show you how they work. So these receptors have a binding site for a signal, so it's going to be this TR, that's the signal, and then normally the channel is closed, but as soon as it receives the signal, it opens up and an ion passes through the membrane. So, and you can see this activity using electrodes here. So scientists can use an electrode to start seeing the activities of these channels. So you see, as soon as, so this is channel closed, and then it binds and then opens and the ion passes. So it's a very fast response. It's the ion channels receptors, they produce a very fast response. They bind to their target and then they allow the ion to pass. When ion passes through the membrane, it generates some sort of response. Then we have receptor enzymes that they use a second messenger. So there is the first signaling molecule that comes in, binds to the enzyme, and then the enzyme does some reactions to generate a second molecule that's going to be the second messenger, and then that second molecule sends a signal and makes something happen. So these receptor enzymes have particular characteristics. They usually have a domain, we call it, a part of this protein that is sitting outside of the plasma membrane, which receives the signal. In this case, we're looking at a receptor tyrosine kinase. And a kinase is a molecule that adds phosphates to other proteins, and usually it adds it to a tyrosine amino acid. So in this case, this membrane-bound receptor is in two units. They're sitting separately, but when the signal comes in, these receptors come together, and then through their internal domain, the part of the enzyme, receptor enzyme, that is inside the membrane, so the signal is received by the part of the protein that is sitting outside, but then the enzyme is inside the membrane. So this particular example, the two subunits come together, and then the tyrosine kinase activity, that enzymatic activity, is activated. And then in this case, the receptor phosphorylates itself. It adds phosphates to its own tyrosine residues. And when that happens, specific proteins come and bind to these phosphates. There are proteins that are sitting in the cell that only can bind to these receptors when these phosphates are here. So these proteins, let's imagine that we have proteins in here. These proteins are going to come and they recognize these phosphate, they bind to it, and then they go ahead and generate a cellular response. So this is a receptor enzyme combination. An example of this is the insulin receptor that you met before when we were talking about molecules, big molecules entering into the cell. If you remember, one of the examples we gave was how cells enter, how glucose enters the cell. And this happens when insulin binds to particular receptors. So this is an insulin binds to the insulin receptor, and this insulin receptor is a tyrosine kinase. It sends a signal as a result of that kinase activity. It sends a signal to these vesicles that have the GLUT4 receptor, the glucose receptor, kind of on a hold pattern in these secretory vesicles. So when the signal comes from the tyrosine kinase, then this vesicle travels to the cell surface and allows glucose to come in. So that's an example of one tyrosine kinase receptor, how it receives signal and the response that it generates. Another type of receptor are G protein coupled receptors. So what does that mean? So you have this receptor, and there this protein, which is called the G protein, and it has 3 subunits. And the alpha subunit of this G protein is bound to GDP, guanine diphosphate. Guanine is a nucleotide, except it has 2 phosphates in it. So normally at rest, this G protein is sitting bound to the G protein coupled receptor. But then the receptor receives a signal, and as a result of this signal, what happens, this alpha subunit exchanges GDP for GTP. So now the alpha subunit is bound to guanine triphosphate, which has 3 phosphates instead of 2 phosphates. As a result of this, the alpha subunit can leave this complex. It can leave this complex and go and generate a response by binding to other proteins. So in this case, the second messenger would be this alpha subunit. And once this is done, once the alpha subunit does its job, then the GTP is hydrolyzed to GDP again, which brings back the alpha subunit to the alpha, beta, and the receptor that it's bound to. So that's a G protein coupled receptor. An example of a G protein coupled receptor is the receptor for the hormone epinephrine, and you're going to meet epinephrine quite a lot. So it is a G protein coupled receptor. This is the beta adrenergic receptor for epinephrine. When epinephrine binds to the receptor, then the G protein dissociates from the receptor, the alpha subunit, although this detail is not shown in here. The alpha protein is going to bind to GTP, exchange GDP for GTP, and then it's going to go and activate another enzyme called adenylyl cyclase, which turns ATP into cyclic AMP. It's a cyclic molecule. So this cyclic AMP is another signal molecule which then goes and makes something happen. Something happens in the cell as a result of formation of cyclic AMP. And once cyclic AMP has done its job, then an enzyme breaks it down into AMP, which then is going to form ATP again later on.